Assalamu alaikum. Today, our topic of discussion is the histology of the thymus gland. So let's start with this topic, which is important. As you all know, the thymus gland is a lymphoid organ. Our learning objectives for today are that at the end of the session, you should be able to identify and describe the histological features of the thymus with functional correlation, and also correlate these histological functions with the clinical condition. So first, let's talk about the immunity, and let's talk about the lymphoid system generally. So how do we define immunity? What is immunity? In simple words, it is the defense system of the human body. And we know that it consists of various lymphoid organs and tissues which have the important function of protecting the human body from the microorganisms, other foreign invasions. The immunity of the human body is broadly classified into two basic types. These are the innate immunity and the adaptive immunity. First, let's talk about the innate immunity. Innate immunity is the non-specific pre-existing defense system of the body. It forms the first line of defense. And this includes many different structures. For example, let's say the physical defense, which is the barriers such as skin and mucous membrane. They provide the barriers to the entrance of the invaders, microorganisms. It could also include the chemical substances. It could include low pH, which destroys microorganisms. Also the secretory substances such as lysozyme, interferon, and thiocyanate in the saliva, which neutralize the cells. The cells such as the natural killer cells, the neutrophils, the macrophages, they're also parts of this innate first line of defense. Then comes the second part of immunity, which is called the adaptive immunity. Now, adaptive immunity is the specific type of immunity, and this comes into action when the non-specific defense systems of the body have failed. This means the adaptive immunity is the second line of defense of the human body against invading microorganisms. The adaptive immunity targets the specific invaders when there is the initial exposure of a foreign antigen or agent to this adaptive immunity. This causes a series or cascade of events which eventually leads to the formation of immune memory. This immune memory provides an acquired resistance to the invading microorganisms. And this adaptive immunity can be further divided into two parts. That is the humoral type of immunity, which involves the antibodies, or it could be the cell-mediated immunity, which involves the action of the cytotoxic T cells. Okay, so since we were talking about the lymphoid system, which is the essential part of the immune system of the human body, we know that lymphocytes are formed in the primary or central lymphoid organs. Now, what do we mean by primary lymphoid organs where the lymphocytes are formed? This includes the thymus and the bone marrow. Then the secondary, or also known as peripheral lymphoid organs, these are the ones where the lymphocytes are activated and their proliferation takes place. This includes lymph nodes, spleen, and also the mucosa-associated lymphoid tissue. Mucosa-associated lymphoid tissue is present in the respiratory tract, the gastrointestinal tract, the urinary tract, right? Okay, now let's come to our topic of discussion today, which is the thymus gland. The thymus gland, as I mentioned before, is the central lymphoid organ, and it is a lymphoepithelial organ present in the superior and the anterior part of the inferior mediastina of the thorax. So here, you can see that this here, if you could just focus on you can see the laser pointer or the red dot over here. This is showing us the thymus gland, and this thymus gland is present over here in the adult or the grown-up within the mediastinum. In which parts of the mediastinum? The superior and the anterior mediastinum of the thorax. Let's now look over here at this thymus gland. If you just follow the red pointer, it is a bilobe structure. Here are the two lobes, the right lobe and the left lobe, and they are connected by this intervening tissue in the middle. And you can also see that the lobes are subdivided into these several lobules. The thymus is a gland which is fully formed and functional at birth, and it is very large and active until the time of puberty. But after that, this undergoes atrophy or slow involution. Microscopic structure of the thymus gland is such that it is a bilobe organ, as I showed you before. Each lobe is covered by the connective tissue capsule. And from that capsule, the septi or trabeculae, they pass inwards and they divide this lobe into smaller sections known as lobules, each being about 2 millimeters in diameter and having an outer cortex and an inner medulla. And each lobule has an outer darkly stained cortex in comparison to an inner lightly stained medulla. This is because there is a greater density of lymphoblasts and small lymphocytes in the cortex in comparison to the medulla. Okay, so over here, when we are looking at the photomicrograph of the section of the thymus gland, and this is the infant thymus. So you can see it's a well-developed gland stained with H&E, and we're looking at it under low power. We can see over here, if you just follow this red laser pointer, this is the capsule which is covering the gland externally. It's a capsule of connective tissue. From this capsule, inwards are extending these septi. They're also indicated by the black arrows. And these septi then divide this lobe of the thymus into lobules. So here is a section which is a lobule, and adjacent to it, here is the other lobule. And if we look at a single lobule, within this lobule, we can see the outer area is stained darker in comparison to the inner part, which is stained lighter. The outer part is stained darker because it has a larger density or concentration of lymphocytes and lymphoblasts. This is the area known as the cortex, indicated by the yellow arrowhead above, whereas the yellow arrowhead below is pointing towards the medulla, which is the inner lighter stained area. Okay, so in this diagram, we have a comparison of the thymus of a child with the thymus of an adult. So in the left side, you can see the child's thymus is well developed with these cortical and medullary regions. Whereas if we look at the micrograph of an adult's thymus, it is mostly atrophied, being replaced by adipose tissue. An important point to remember in comparison to the lymph nodes is that in thymus, there are no lymphoid nodules. And also there are no afferent lymphatic vessels. Only a few efferents are present in the septi. Stem cells, remember, are formed in the bone marrow, and from there they travel to the thymus. And when they reach the thymus, they first settle in the superficial part of the cortex, and then they divide repeatedly and give rise to the T lymphoblasts and the small lymphocytes. And these lymphoblasts and small lymphocytes are also known as thymocytes. The medulla of the thymus also contains lymphocytes, but they are less densely packed. And over here, the epithelial reticulum is more obvious. We're going to talk in a little detail about the epithelium reticulum in the next few slides. Remember that the mature T lymphocytes exit the medulla eventually by passing through the walls of venules and efferent lymphatics in this region. Now let's discuss the important cell type, which is the thymic epithelial cells, abbreviated as TECs, also known as the epithelio-reticular cells. These are the cells which are embryologically derived from the endoderm, and they are the cells which have a large euchromatic nucleus, and they are morphologically and functionally diverse. This means they have a wide range of functions. And these cells, the thymic epithelial cells, they join to form sheets that cover the internal surface of the capsule of the thymus, as well as the surface of the septi and blood vessels. And this also leads to the formation of the blood-thymus barrier. This is a barrier which prevents unregulated exposure of thymocytes to the antigens. So if we would enumerate three components of the blood-thymus barrier, they would be, number one, the endothelium of the capillary wall with the underlying basal lamina and pericytes. The second component would be macrophages, and third would be the type 1 epithelio-reticular cells. What about the type 2 epithelio-reticular cells? These are lying in the cortex, and they have processes, they have long extended processes which join similar processes of the other type 2 epithelio-reticular cells to form a network within the thymus known as the cytoreticulum. What is the significance of this cytoreticulum? The macrophages and the developing lymphocytes within the thymus gland, they are attached to this cytoreticulum. Okay, so to elaborate and explain these bullet points, we have this micrograph on the left, and over here, this has been taken from the cortex of the thymus. Now, in the cortex, of course, the cell population that we initially talked about, we can see the lymphocytes and the lymphoblasts. So there are many lymphocytes and many lymphoblasts over here. Plus, there is another cell population which is comparatively lighter stain. You can see over here these large-sized epithelial cells. They have a pale staining, larger nucleus. These are the thymic epithelial cells, also known as the epithelio-reticular cells that we were just talking about. Now, these cells are difficult to visualize in the routine H&E stain. So we use special stains, such as in this case, the PT stain has been used to make it easier to visualize these epithelial cells. So because of the large number and density of lymphocytes, these epithelial cells become obscure. Anyhow, when we look at this diagram on the right, we can see that these are the epithelial cells with a large nucleus and long processes. You can see the long processes of adjacent thymic epithelial cells are joined or attached to each other by this cell junction, which is the desmosome. So this creates a network known as the cytoreticulum. Reticulum literally means network, and cyto means cells. So this is a network of cells created within the thymus by the thymic epithelial cells. And on these processes and on this network is the attachment of, you can see these lymphocytes. So the lymphocytes and also the macrophages, they're attached, or you could say they're anchored. They're anchored to this cytoreticulum. This is a unique feature of the thymus gland. That's a very important point I'm about to mention, that better to mention in the next slide. Keep it in mind that the supporting framework of epithelio-reticular cells, this is a unique and unusual feature of the thymus gland. What's the advantage of having a network of the thymic epithelial cells instead of the simple network of reticulin fibers, which is seen in the other lymphoid organs? Well, the advantage of this is that this cytoreticulum formed by the epithelio-reticular cells, which are APCs, that is, they are the antigen-presenting cells, allows regulated specificity of lymphocyte binding. Now, since we have mentioned a few types of epithelio-reticular cells, let's talk about the type 5 epithelio-reticular cells, which also form a cytoreticulum in the medulla. Whereas type 3 and type 4 epithelial reticular cells, they occur at the boundary of the medulla and the cortex, and including junctions between their sheet-like processes form functional corticomedullary barrier. The thymic epithelial cells, they, like I mentioned before, act as antigen-presenting cells, and they also secrete cytokines for T cell development. So they're also known as the thymic nurse cells. Now, another important and distinctive feature of the thymus gland, when we talk about the histology of thymus gland, are the Hassel's corpuscles. What are the Hassel's corpuscles? These are small rounded structures about 100 microns in diameter, and they're located in the medulla of the thymus. They are formed by concentrically arranged type 6 epithelial reticular cells. And they represent a degenerative process, but they are not entirely non-functional, as research has shown that they also produce cytokines. Now we see a photomicrograph in front of us. On the left, we can see in the low power that the human thymus has this external part, which is cortex, which is darker staining in comparison to the inner lighter stained medulla. So this is the hematoxylin eosine stain, and it is showing us the outer cortex and inner medullary region of the thymus. Externally, you can see the eosine stained connective tissue capsule, and also the septum or trabeculum, which is extending from the capsule inwards. We see that this medullary region has some light pale staining eosinophilic structures, and these prominent structures are known as the thymic or Hassel's corpuscles. Now, on the right side, at a higher magnification, let's focus on this prominent structure over here, which is the Hassel's corpuscle. So as we can see, this is consisting of the masses of closely packed, concentrically arranged type 6 epithelial reticular cells with flattened nuclei. And in the innermost part of this Hassel's corpuscle, there are the fully keratinized cells. The rest of the medulla is packed with these lymphocytes, which have the basophilic small nuclei, as you can see, whereas a few cells which over here are marked by these, pointed out by these black arrows, these are the cells which are the cells which are epithelio-reticular cells forming the reticulum, the cytoreticulum within the medulla. So these are the type 5 epithelio-reticular cells, and they have a large pale staining nucleus because of the euchromatin. Okay, so let's summarize the functions of the thymus. We know it is involved in development of immunocompetent T lymphocytes. Also, proliferation of clones of T cells occurs over here to supply the peripheral tissues and circulating pool. It also secretes polypeptides such as thymosins, thymolin, and thymopoietin. This is a very important point that I would like to mention and discuss when we talk briefly about the function of the thymus. The thymus is the site of self-education. Now, what do we mean when we say self-education? It means that there is going to be development of central self-tolerance, and there's going to be apoptosis of the self-reactive cells. Now, what do we mean by self-tolerance? We know the thymus, it is producing these lymphocytes which are involved in the defense system of the body. We don't want the defense system of the body to target the self-antigens. And this process of creating an awareness within the lymphocytes of not to attack the self-antigens or the cells of the body itself is known as self-education. This is a process which occurs in two stages. The first stage is positive selection, and the second stage is negative selection. What happens in positive selection? First, let's talk about where is it taking place. It is taking place in the cortex of the thymus gland. So in the cortex of the thymus gland, the process of positive selection occurs. This is the process which allows survival only of those T cells which have functional T cell receptors that will recognize the major histocompatibility complex molecules. Now, we have mentioned the MHC or major histocompatibility molecules, class 1 and 2. These are the molecules which are responsible for identifying and bringing the invaders or the intruders, the microorganisms to the T cells. And T cells cannot recognize an intruder unless it is marked by the MHC molecules. So in order to be functional, the T cell needs to recognize the MHC molecules. In a very simple flowchart over here, if you follow the laser pointer, you can see that this is a pre-T lymphocyte, and it has T cell receptors, abbreviated as TCR. Now here it is brought into contact with this, you can see the cortical epithelial cell that has the MHC molecules. Now, if this pre-T lymphocyte attaches and recognizes the MHC molecules, that means it binds to the MHC molecule. Only in that case will it survive and then move inwards into the medulla. If it does not attach to the MHC molecule, then it will be gotten rid of by programmed cell death, that is apoptosis. So this is the positive selection. And those cells which have passed through the process of positive selection and moved into the medulla, then in the medulla, they undergo the next process, which is negative selection. Now, negative selection means that there is going to be survival only of those T cells that do not attach or bound to the self-antigens. So here in this flow chart, we can see that we have the pre-T lymphocyte in the medulla, and here is the antigen presenting cell which is presenting the self-antigens. Now, if this pre-T lymphocyte recognizes, by recognizing we mean it attaches to, it reacts with the self-antigen, in that case, it is going to be gotten rid of by apoptosis. And if it does not recognize a self-antigen, only in that case will it survive, and then its maturation will be completed. And now we say that it has passed through the process of negative selection, and now it is immunocompetent. Immunocompetent meaning now it is ready to fight the invader or the intruder, and it knows that it is not going to destroy or attack the self-antigens. Keep this in mind that 2% of cells survive the selection process in the thymus. So this is such an elaborate and important process taking place that initially the lymphocytes from the total population, it is just 2% which survives the whole selection process. And eventually the lymphocytes, the T lymphocytes that are ready to invade, we could say attack the invader, or these are now the naive immunocompetent T lymphocytes, and they can either be the helper T lymphocytes, which retain only the CD4 protein, or they can be the cytotoxic T lymphocytes, which retain only the CD8 protein. Okay, so this concludes with the description of the histology and the function of the thymus. Medically, there is this condition that I would like you to be familiar with, that is the congenital condition, which is known as Dye-George syndrome. This occurs due to failure of the third and fourth pharyngeal pouches to develop normally within the embryo. The third and fourth pharyngeal pouches, if they do not develop normally, there is going to be aplasia, that is the absence of, or hypoplasia, which means underdevelopment of the thymus gland. And if the thymus gland is underdeveloped, we know that the T lymphocytes will not be produced properly, and obviously the immunity will be affected, and this causes the higher susceptibility to infections in the individuals suffering from Dye-George syndrome. So at the end of this topic, just a few MCQs I would like to share with you to retain some important aspects, some important points. So you know regulated specificity of lymphocyte binding due to a reticulum of epithelial cells is a unique feature of which tissue? We now know it is the thymus. Many immune-related cellular activities are often impaired in aged patients. the lymphoid organ, which normally develops less functionality and increasing amounts of adipose tissue with age, is the thymus. So this is a simple micrograph, and the organ which is being shown to you is the thymus, and 2 points towards the cortical region. 3 is showing the comparatively lighter stain medullary region. 4 are the connective tissue septi. And 5 is also a thymus, but this is a thymus of an older individual. It is, you can see, involuted, right? Another diagram of the thymic cortex you can see above with these thymic epithelial cells, that is labeled 2 in the diagram as well. Whereas 1 points towards the lymphocytes in the micrograph as well as the lymphocytes in the diagram and the micrograph below. And number 3 is pointing towards this eosinophilic prominent mass in the medulla, which is the thymic corpus. So above we have the thymic cortex and below we have the thymic medulla. Okay, so this concludes with today's discussion topic, which was the histology of the thymus gland. And the lecture source, I have shared the books with you from which the micrographs were taken. And if you like this discussion, if you like this lecture on the histology of the thymus gland, then please like and subscribe to my YouTube channel, which is AnatomyMentor. And you can do that by simply clicking on my picture shown at the end of this video. And you can also follow my Facebook page, which is AnatomyMentor. Thank you and take care. And please follow me for further lectures, also on embryology, gross anatomy, as well as neuroanatomy. Thank you, take care, and Allah Hafiz.